Good afternoon. This is Dr. Daniel Guerra coming to you from Authentic Biochemistry Studios in the Inland Pacific Northwest. Today, of course, is the 9th day of February, and the year is 2021. So, I'm going to give you a video lecture today rather than my normal audio, which I periodically do anyways because often it's more illustrative because I want to show you some of the principles, and that's best shown often by diagrams and cartoons rather than just by describing it verbally. So that's one of the reasons. The other reason is because I like to get to know my audience better, and sometimes the visual contact is more useful. So that's what we're doing today. So let's get started. Again, my name is Dan Guerra, and I have a Ph.D. in biochemistry and physiology. I've been a professor of biochemistry for about 30 years, and I've also been a research scientist. I've worked in lipids, I've worked in neuroscience, I've worked in pharmacology, and just basic lipid metabolism. So those are my credentials. If you haven't watched me before, I've been teaching. I had taught for several years, as I said, graduate level biochemistry as well as specialty courses, DNA metabolism, vitamins and hormones, and then a lot of my classical guest lecturing was done in lipids, lipid biochemistry, lipid pathophysiology. So that's my background. And what we've been doing in this arc of lectures ever since really around, before Halloween, so that puts it, 4 months back, I've been working on the aging lectures, which is a revitalization of a whole semester course I taught some years ago at Gonzaga University, which was a graduate course in biology and physiology of aging. So I've revamped all those lectures, and what I'm doing now is bringing you up to date with all of the current literature that describes the aging process in humans, the morbidity that's associated with it, and then ultimately, what are the major causal events that lead to mortality. And of course, those are going to be plenum and florid, but I'm trying to get at some of the background prodromal stages, and those, of course, involve pathophysiological diseases. So that's where we're at. So today I'm going to talk about, in a generic sense, aging and dyslipidemia, but you'll see that it gets very specific very quickly. So a paper published about, almost 2 years ago now, in Nature Reviews, was in gastroenterology and hepatology. And it tells us the following, and it's nothing I think that most of my listeners don't already appreciate. Obesity is an epidemic. About 2 billion adults worldwide are overweight or fat, according to the World Health Organization. And it isn't enough to start keeping your eye at a 6'1" level or above, because you have to add over 40 million kids 5 years and younger to do that. So rather than just avoid noticing that obesity is really common, I'm trying to deal with it and discuss it often in my lectures. And I've been doing this for a number of years, actually probably since the early 90s. There are obvious links of obesity to cardiovascular disease and to a major disorder that can lead to a carcinoma known as non-alcoholic fatty liver disease, or NAFLD. And so the paradigm in that disorder is it goes from obesity to this non-alcoholic fatty liver disease to hepatocellular carcinoma. And we've talked about this in the past. So obesity is becoming, I would say it has become now, the number one health-related physical abnormality pathology, and it's dangerously increasing in younger and younger populations. Even children preschool now are becoming more and more diagnosed with being overweight or indeed frank obesity. So what is obesity normally described as? Well, it's defined as having a body mass index equal to or higher than 30 kilogram per meter squared. Now that's a metric that's used. But remember that if you're a muscular person, or you put on adipose in different locations other than the viscera or certain other components of the trunk of the body, and you're either male or female, and you have other kinds of predisposals, such as, again, high level of skeletal muscle development, that BMI can be quite off. So you can have a BMI that's over 30 and not really be a person that's obese. Many people I know that work out or that are very athletic have that BMI or greater, and they're not obese. So it's a metric that's used, and maybe it's better than just live body weight or mass, but it's not without its fault. But what's correlated with obesity? And when you see it, you know it, right? A metabolic syndrome, which we've talked a great deal about in my lectures, basically hypertensive problems associated with the cardiovascular system, maybe atherosclerosis. The obesity, of course, is a linked factor there. Metabolic syndrome is often prodromal for the next major pathophysiology, which is type 2 diabetes. Both metabolic syndrome and type 2 diabetes are associated with dyslipidemia. That is, an alteration in circulating lipid profiles that occur over time, chronically, and ultimately induce multiple pathologies, including necrosis on one end, cellular death, that is. On the other end, even an increase in cell proliferation and tumorigenesis. And then every other kind of possible malfunction of cell fate between the two. Okay? And that leads to the third major component that you get. This is this NAFLD, which I've talked about. And then also there is simply an inflammation of the liver known as steatohepatitis. That's called NASH. Again, these are both the non-alcoholic varieties. There is a very powerful alcoholic senescent process in the liver, and that is well described in the literature. Primarily, I'm going to stay away from that because I talked about it in the past. I'm talking here more about obesity linked, not alcohol linked. But the two can overlap quite a bit, actually, especially in the West. Obesity, of course, is a major risk factor for several common types of cancers I just mentioned. Most notably of that is the pancreatic and hepatic cancers, but you also have various types of gut cancer, and even brain cancer has been linked to obesogenic state. And we talked about this also in the past. There's some of the most lethal cancers that you find in humans and what takes people down. And the survival rate is 5 years, thereabouts, right? And that's of a rate of about 4 to 8%. Hepatocellular carcinoma is the most common form of liver cancer. And the connection between obesity and that cancer is mediated by a state of chronic, low-grade inflammation in the involved tissues, in this case, liver. So high-fat intake, but increasingly became aware in the 90s because of lipid biochemists, such as myself, trying to get the AMA to look at carbohydrate contribution to obesity. But the high-fat research is mostly in the murine model. There's the rodent model. But in humans, a lot of the obesity is caused by excessive carbohydrate in the diet. Lipid metabolism has very stringent feedback control mechanisms because it's lipoprotein mediated peripherally, and that means that the circulation to the adipose and then back from the adipose to the rest of the body, where it can cause pathology, such as in the heart, even in skeletal muscle, and certainly in the solid organs like the liver and the pancreas, is a process that has a lot of delimiting, overriding feedback regulation, whereas carbohydrate metabolism doesn't have that kind of delimited control because it's primarily controlled by insulin and insulin insufficiency caused by the lack of sensitivity to insulin binding to its receptor. And all that has to do with chronic nutritional overeating, and that overeating can be from calories from any of the major food groups. but primarily that comes from carbohydrate. So I want to make sure that that's established. So high intake of calorically dense foods, it can be from lipid, but primarily it's from carbohydrate in terms of percent. You can get endocrine disorders, which we've talked about great deal. We just were speaking about adipokines, so I told you something about that. Sedentary lifestyle, there's a lack of exercise throughout life. The earlier you stop exercising, the more likely you'll get obese. There are hereditary factors, but they don't really contribute to the gross obesity that we see massively increasing in the United States as well as in the rest of the world. And some medications can cause it too. For example, some neuropsychiatric medications can induce an obesogenic state in the short term, but usually that gets reconciled by normal metabolic feedback control. And there are also, of course, the utilization of cortisol and cortisone. These are used as anti-inflammatory drugs, often pre and post-surgery, for example, but increasingly for a lot of disorders that are, in fact, obese-driven. Okay? Those can also enhance adipose deposition, again, usually in the short term because people aren't on these drugs chronically. Although the use of corticosteroids for decreasing inflammation has become ubiquitous, and it's a highly prescribed pharmaceutical, and that has been linked to some chronic manifestations of an increase in lipogenesis and therefore obesity. But again, this is not the major cause. The major cause is excess calories, primarily from carbohydrate, but also from lipid. Can, of course, be from protein too. And then the sedentary lifestyle. So what it leads to then is this kind of like downward spiral. It can cause obesity, and obesity can increase the amount of reactive oxygen and reactive nitrogen that's synthesized in all cells. That can cause DNA damage. Obesity can lead to non-alcoholic fatty liver disease. It can lead to non-alcoholic steatohepatitis. NAFLD can also just cause hyperplasia of the liver, primarily as they're fibrocytes. NASH can lead to fibrosis, and fibrosis leads to cirrhosis, which is the deterioration of most of the cells in the liver, particularly the hepatocytes. And all of that then can lead from hyperplasia directly to hepatocellular carcinoma, or from cirrhosis to HCC. And of course, enhancement of DNA damage can cause that disease to progress. Key points here is there's a steady increase in the beta and the BMI, and that's become a worldwide pandemic. It's currently estimated to cause more than 90,000, I'd probably put that over 100,000 cancer-related deaths per year in the United States alone, because this is older data here. Although HBV and HCV, those are viruses, they're hepatic viruses, right? Those infections are considered a major risk for hepatocellular carcinoma. At least in the United States, obesity is far more likely to be the major risk factor for generating HCC. And of course, that's along the road from metabolic syndrome and type 2 diabetes. Obesity is implicated in the genesis of insulin resistance. It's a major phenotype, pathophenotype, biochemical phenotype, if you will. You also have that linked to type 2 diabetes, as I said. You can find it in non-alcoholic fatty liver disease before there was any kind of diabetes diagnosed, and NASH, of course, inflammation of the liver because of fatty deposition. You also get hepatic fibrosis, as I said, and then cirrhosis, and all of that can result in further serious complications, not just in the liver, but in the rest of the body. And that can, of course, give you liver failure and then full-blown cancer. Obesity is associated, of course, with chronic low-grade systemic inflammation, which involves adipocytes, which we've been talking about when we talk about the adipokines, but also in the rest of the body, including the central nervous system. And the immune cells themselves become hyperpolarized and pro-inflammatory because of the secretion of cytokines and chemokines, growth factors, etc., that can lead to an overall systemic inflammatory chronic state, all of which can lead to more morbidity, resulting in an inflammatory response linked to DNA damage, proteinopathies, and of course the massive increase in lipid, both intracellular and extracellular, which can auto-oxidize and further generate more reactive oxygen, inducing the stalemate destruction of all of those macromolecules I just mentioned, for example, DNA, RNA, and polypeptide. So you get hypertrophic adipocytes, that's one of the hallmarks. And what they do is secrete fatty acid. Free fatty acids are toxic because they don't normally move through the membranes, through the channels that they would typically move through, but rather they can coalesce into membranes and disrupt those membranes. It basically cause disruption in ionic gradients and therefore polarization of the membrane, and all of the normal signaling that the cell can do because of that becomes corrupted, further disrupted, and that can lead to a hyperimmune response because of the presentation of polypeptides on the surface of even epithelial and endothelial cells, polypeptide fragments that are generated because of an overload of proteinopathy, leading to the unfolded protein response, causing degradation of glycoproteins that would normally be selectively secreted from cells. Immune cells and also almost all other cells secrete some component extracellularly to interact with the extracellular matrix, and some of these can be metalloproteinases, and of course there can be exosomes generated, which can have microRNA, further then translocated to other cell lineages, coalescing in the membranes that have been corrupted because of dyslipidemia, and then inculcating and interfering with gene expression in those cells where those exosomes have now been taken into the endosomal pathways, where those microRNAs can then corrupt translation of certain proteins. And that can then lead to malfunction of chromatin remodeling, DNA damage repair. It can cause those cells, again, their fate to either go into autophagy and stay in autophagy, which then can result in the accumulation of mutations leading to oncogenesis, or it can lead to full-blown programmed cell death, the various types we've talked about, apoptosis, necrotosis, ferroptosis, etc. And that can lead to further inflammation, or if nothing else, neurodegeneration or just cellular degeneration in important tissue beds and vascular beds that then become chronically disabled and dysfunctional, and therefore leading to morbidity. All of this is exacerbated as you age because of the amount of reactive oxygen that's made, but also because of the fact that cells pick up mutations. Okay. Now, at the end of this particular slide here, it'll tell you that there are various pro-inflammatory cytokines. These are some of the classic ones. I know that most of my listeners are well aware of these, but TNF, necrosis factor. The interleukin 6 is an important one, interleukin 1 beta, of course, which has to have a proteolytic processing before it gets synthesized and secreted. Interleukin 8, 10 can sometimes be anti-inflammatory depending on receptor-mediated responses. 18, of course, interleukin 17 typically come from Th17 cells, but can be secreted from other, not only immune cells, but from other cells in the system. And then there are a whole series, again, of adipokines. We've talked about leptin and adiponectin, for example. Hyper-adiponectemia, hyperleptinemia, or hypoversions of both of those alterations of adipokines in the bloodstream, therefore not correctly firing in the arcuate nucleus of the hypothalamus, dysregulating the feeding response, which can exacerbate then the obesity. We talked about that just recently. In fact, that's why I'm doing this lecture now. This is following what we've been talking about. So there's an axis of disease progression. NAFLD presents over several years, obviously. It's characterized by progressive accumulation of hepatocytic triglycerol. It represents a pathophysiological spectrum of massive TAG accumulation and steatosis that would again be an inflammatory response in the liver. TAG accumulates more, you get also inflammation, injury, and directly then generating steatohepatitis, and not just this NAFLD, okay? So you get both aspects, just lipid deposition, but also hyperinflammatory response, which is basically an immune response that has nothing to do with pathogen, right? So it's called sterile hyperinflammation, excuse me. Okay, anyway, that can progress to hepatic fibrosis. This is production of fibrocytes rather than hepatocytes because of the loss of functionality of cell division. And these fibrocytes don't act as hepatocytes because they don't have the right gene expression pattern. So they're a null set of cells which can go through various stages of alteration, leading even to oncogenesis. And that's what happens when HCC kicks in. That's the tumorigenic tract. Current global NAFLD is probably at about 25%, which is an astonishing increase from 15% just in 2005. And it was up to 25% in 2010. It's probably about 28 to 30% in 2020, okay? Although numbers vary and they go up and down slightly. That's about where we're at. Almost, again, about very close to a third of the population can somehow eventually generate an NAFLD during aging, okay? So it's linked to metabolic syndrome, type 2 diabetes, and as I call it, constellation of a lot of somatic and central nervous system associated pathologies, and they're all linked to dysfunctional bioenergetics, right? We talked about this many times in lecture. Signaling failure, extracellular, intracellular signaling, and then, of course, the pathological presentation arising from neuroimmunoepigenomic modification of gene expression. So the corruption of the normal fine-tuning of gene expression because of epigenetic modifications of chromatin during chromatin remodeling, such as methylation patterns, acetylation patterns, etc., and histones and on cytosine residues and promoter regions, enhancer regions of certain genes, then corrupting not only the gene expression but the splicing of particular exons, which can form polypeptides that can be even under their own guise pathogenic. Okay. So let me move myself out of the way here. Sorry. Influence of long-term treatment with sucrose and drinking water on the kidney histology. This is a mouse study. An inflammatory gene expression and lipogenesis using male Wistar rats supplemented with 30% sucrose. Now, sucrose is a carbohydrate that is very common in the human diet. That's why they use that rather than glucose. Remember, it's a disaccharide of glucose and fructose. It's a plant sugar, right? So you've heard of high fructose corn syrup. Well, the fructose comes from sucrose. Sucrose is the translocatable sugar in higher plants, as opposed to glucose, which of course is a translocatable sugar in animal tissues, right? At least homeothermic animals. Okay, so there's a kidney histology associated with this, and you get it with sucrose treatment, and this is a rat study. You get increased renal lipid content. So we just talked about HCC, now we're talking about kidney. And you get, because of that, glomerulosclerosis, right? That is the sclerosis of the glomerulum, which is that component of the kidney which does the filtration process from that organ. You get TGF-beta, and you also get TNF-alpha gene expression. Both of those can contribute to inflammatory responses as transcription factors or working through the receptors, respectively. Sucrose feeding is associated with increased saturated and monounsaturated fatty acids and a decrease in the C18 essential fatty acids, which are linoleic and alpha-linolenic acids, 18:2, 18:3. 18:2 is omega-6, 18:3 is an omega-3 fatty acid. Those are your two essential fatty acids in the diet. Those then lead to elongation and further desaturation to the really important very long-chain polyunsaturated fatty acids, such as arachidonic acid, which is the omega-6, classical omega-6 VLC, PUFA, very long-chain polyunsaturated fatty acid, and then, of course, eicosapentaenoic, that's 20:5, or docosahexaenoic, that's 22:6, which are of the omega-3 class. And we talked a lot about that in a lecture of the last year, easily. All right, so the content of arachidonic and docosahexaenoic, that's omega-6, omega-3, did not decrease despite the decrease in the content of their precursors. That would be then those 18 carbon precursors. And that has to do with poor lipid absorption, as well as dietary issues, right? Neither the expression nor the protein quantity of a delta-5 desaturase, don't worry, I'm going to show you where that works in the metabolism in a moment, increased, okay? Indicating the renal delta-5 desaturation is not important for the preservation of the kidney arachidonide and docosahexaenoic acid content in metabolic syndrome, which is what they were looking at here. Now, I have a question about that. I'll show you why in a minute. The expression of the sterile response effector binding protein 1C, which is a transcription factor, increased twofold in the metabolic syndrome group, indicating an increased renal lipogenesis, because that's what that transcription factor is responsible for, cholesterologenesis and fatty acyl synthesis both, in the sucrose-treated rats. Now, because sucrose generates fructose upon the sucrase enzyme, what you generate then is fructolysis rather than glycolysis. And when you get fructolysis, directly making fructose and go through fructose 1,6, first fructose 6-phosphate, or fructose 1-phosphate, then fructose 1,6 bisphosphate, because of the second kinase, you are overriding the Pasteur effect of the control over PFK1, PFK2, which has to arise from the isomerase from glucose 6-phosphate to fructose 6-phosphate. That's under metabolic control. So fructose isn't under normal metabolic control. That's why it's obesogenic, by the way, sucrose, and then vis-a-vis ultimately the end product fructose after that disaccharide is broken down. But also it seems to be directly associated, this is what they're looking at here, with transcriptional regulation of lipogenesis in the kidney in this state. And that, of course, is linked to metabolic syndrome because of the hypertension. That's why they're looking at it in this model, the mammalian model. So chronic sucrose intake induced, in this model, glomerulosclerosis, low-grade inflammation, and increased saturated and monounsaturated fatty acids, lower the C18 essential fatty acid content, but it did not seem to alter the elongation to the AA or DHA fragment in this animal model. Now, there's a lot of problem with that, extrapolating that to humans, because our desaturation pathways, our lipogenic regulation is different from rats and mice, substantially so at the level of transcriptional regulation. I want to remind you a little bit about this. Now, the role of the liver and adipose tissue in digestive lipid metabolism. What does the liver do? Well, it does many things, but I'm just giving you, so that you'll recall. I know the physicians already know this, and the scientists that are listening to me know this, but a lot of other people, maybe they haven't thought about it lately, or they don't know exactly what these organs do. This is just a very brief overview. If I was teaching physiology, I would spend weeks on liver alone, right? But what the liver does is synthesize, secretes bile salts, which helps in digestion. That's all I'm going to say here. Synthesizes, secretes, and receives lipoproteins, all the major lipoprotein fractions, except for chylomicrons, which are synthesized in the small intestine. And it synthesizes new lipids from non-lipid precursors and transiently stores them. That's the whole gluconeogenic pathway, which is major paradigmatic in the liver. Adipose tissue absorbs triacylglycerol from lipoproteins and cholesterol esters, actually. You can pick it up from chylomicrons directly after digestion from the small intestine into circulation. And it picks up these lipids directly from a protein known as lipoprotein lipase. Stores triacylglycerol, that's what visceral fat primarily is. That's why adipocytes increase in volume, size, but they don't necessarily go through a lot of cell divisions. And of course, the adipose is a major storage organ system. You know that it regulates metabolic homeostasis, because we already talked about very recently the adipokines, right? I did some video lectures on adipokines and many audio lectures, so we don't need to overcome that largesse of information that we already have produced. We could just leave it as it is in its plenum and florid content. So, okay, it also sets, besides being a major organ system for dietary and BMI set points, because the lipid depot fat concentration in the body does regulate not just feeding, again through the hypothalamus, we talked about this recently, but also an overall redistribution of lipids to the periphery, which is a very important component of what morbidities you might pick up, such as cardiovascular disease. And again, on the other spectrum, besides a degeneration of tissue mass or hypertrophy, for example, in the cardiac muscle, you get a very high probability of increasing because of the inflammatory response associated with all this lipid mobilization in generating mutations that lead to oncogenesis and then tumorigenesis. So this gives you a basic overview. You can digest cholesterol esters, phospholipid triacylglycerol is going unchanged. Moving through the digestive system, most of the esters, phospholipids, triacylglycerol, some short chain and medium chain fatty acids bind with bile salts, right? These emulsify these lipids. You also get pancreatic enzymes like lipases and, of course, glycosolases to break down glycolipids. All that's degraded and sent into then the small intestine, where primary products are fatty acids, 2 monoacylglycerols, cholesterol and cholesterol esters, and then you also have remaining fragments of phospholipids. This all moves through the chylomicrons. The chylomicrons then are your first after reesterification, and that's based on the amino acid content after proteolysis from dietary protein consumption. So the amino acid profile of your diet will help control the correct synthesis de novo, that is transcription translation, of polypeptides that end up being apolipoproteins for chylomicron assembly in the small intestine. So the correct molar concentration of amino acids is more linked to animal protein than it is to plant protein. So you can get deficiencies in chylomicron assembly if you're on a strict vegan diet, unless you have mammalian protein as a supplement. This has been known classically for many years. So that's what cholesterol esters are. You have fatty acid. Cholesterol esterase will generate free cholesterol, free fatty acid. Here's a phospholipid like phosphatidylcholine. Lipases will generate glycerol phosphorylcholine, shown here, where those 2 fatty acids are now into metabolic play, or used to reesterify to make triacylglycerol, for example, in the adipose. And there you show directly triacylglycerol can make the 2 monoacylglycerols. This is all happening in the small intestine. Now, a paper published 3 years ago, not quite 3 years, 2.5 years ago, July of 2018, in that journal tells us the following. Essential mitochondrial succinyl-CoA oxoacid transferase, known as SCOT, will convert acetoacetate to acetyl-CoA. Acetoacetate, remember, is a ketone body. Ketone bodies are generated after glycogen stores are depleted. Glucose levels start to drop. This is the utilization of depot fat to make ketone bodies, because free fatty acids cannot go into circulation. And what's most important is that ketone bodies like acetoacetate and beta-hydroxybutyrate, which comes from an NADH-dependent dehydrogenase in the membrane, those 2 ketone bodies can make it to the brain through the blood-brain barrier and be used when glucose levels start to diminish. Okay, so it's very important during long-term fasting and ultimately starvation. So that's what normally happens there. Histone lysine acetylation will diminish chromatin electrostatic interactions between double-stranded DNA and the lysine-rich nucleosomes, and that's a remodeling phenomenon to the chromatin, to a euchromatin from a heterochromatin state that opens for a possibility or probability of allowing for RNA polymerase II to get into the DNA and therefore mediate global transcription. So that's what happens when you acetylate a lysine resident histones. Methylation is more complicated in terms of what it does to gene expression, but ultimately there's also lysine methylation. There's lysine ubiquitinylation, which is not about proteasomal degradation at this point. There's propionylation, butyrylation, 2-hydroxyisobutyrylation, succinylation, here's the first enzyme we talked about, malonylation, glutyrylation, hydroxymethylglutyrylation, crotonylation, and beta-hydroxybutyrylation. So a whole host of alterations on lysine residues and histones, and sometimes nucleic acids embedded in promoters and enhancer regions, which will co-regulate because of chromatin remodeling and then ultimately epigenetic phenomenon, the alteration of gene expression. All of because of excessive amounts of lipid, which can then cause a movement off of the equipoise and then dysregulate normal gene expression, you see. So fatty acyl-CoA during ketogenesis, you get beta oxidation to acetoacetyl-CoA. Acetoacetyl-CoA, because of H&G-CoA synthase 2, that's hydroxymethylglutarol-CoA synthase 2. You add another acetyl-CoA, now you've added 6 total carbons, that is. You'd make hydroxymethylglutarol-CoA. Then there's hydroxymethylglutarol degradation, removing acetyl-CoA. And then you get back to acetyl-acetyl-CoA. Okay? So you started off with the fatty acyl-CoA. You make acetoacetyl-CoA. These are thioesters. They do not move through membranes. This will move through the membrane now, acetoacetate, There's your enzyme BDH making beta hydroxybutyrate, that is. Remember I told you it's an NADH dependent enzyme. That's in the membrane. But acetoacetate, because of that SCOT enzyme, can take succinyl-CoA and make succinate and go back to the CoA thioester. CoA thioester then to 2 acetyl-CoAs because of the thiolase reaction. The acetyl-CoAs in the TCA cycle, of course, through the NADH over DH2 synthesis, make a lot of ATP. But the acetyl-CoAs can also lead to histone lysine canonical acetylation. That will give you, because of this excessive amount of fatty acyl-CoA incorporated, a selective chromatin remodeling and an alteration of transcription. So, once again, beta-hydroxybutyrate and acetoacetate are the primary circulating ketone bodies. There they are. They are increased ketosis and synthesis and circulation during fasting, as I mentioned, starvation, as I mentioned, stress, particularly chronic, inflammatory responses in general because of obesity, and the removal of carbohydrate from the diet. This is, again, the mouse and rat models. You never remove carbohydrate from the diet in humans. So you can get diabetic ketoacidosis if you have too high a level of this, and that's actually paradigmatic for this whole process. Beta-hydroxybutyrate and acetoacetate also serve to control gene expression, and that's what I just showed you there. Epigenetically, via control over histone lysine acetylation. This is something that's now well known to be exacerbated in the obesogenic environment. Now I'm going to tell you about desaturation. So starting off with stearoyl-CoA, which is an 18-carbon saturated fatty acid, and molecular oxygen, you will make oleoil-CoA, which is an 18:1 with a double bond between the 9 and 10 carbons. So it's a delta 9, 18:1, oleoil-CoA. Now this enzyme requires a desaturase in its reduced form, which is a 2Fe2+, to the desaturase in the 3+ or oxidized state, right? Ferrous to ferric. This will then drive this cytochrome B5 from its reduced to oxidized state. It can be re-reduced using enzyme-bound flavonoidenine nucleotide reduced form in the NADH cytochrome B5 reductase, generating the enzyme in the FAD oxidized form, ultimately then taking NADH to re-reduce the FADH to keep this desaturation going, because that's what's going on here. The transport and mobilization of the reducing power here, right? Ultimately, then it's going to use NADH to carry out this final reaction. And it'll generate NAD, which then has to be re-reduced, right? Normally because of glycolysis or maybe fatty acid oxidation itself in some other cell system, right? All right. Now, there's the end of a given fatty acid. And we're going to look at the methyl terminus, which is known as the omega carbon. And then you have a methylene group, another methylene group. So the third carbon is called the beta. I know this is weird nomenclature, but it's how we do it. And the second carbon here is the alpha. And then you have this carboxylic acid in the 1 position. So this N number can be anything, right? It could be, you got 1, 2, 3, 4 besides that. So 18 carbon fatty acid, that would be 14, right? Or if it's a 20 carbon fatty acid, it's going to be 16, right? So what we're talking about is where the double bond is from this omega carbon. So it's 1, 2, 3. So the double bond that we'll call this an omega-3 fatty acid eventually would be between this alpha and this beta carbon. Even if this is highly removed, it depends on where it's happening and what methylene structure, where it is in the sequence of events topologically, right? Numbering, again, from the carboxy terminal to the methyl terminal. So here's an omega-3 fatty acid where you've got, remember, here's your methyl terminus, there's your methylene, and then you've got the double bond between the third and fourth carbon. That would be called an omega-3 double bond. Remember the DHA story we talked about 4 slides ago. So this would be an omega-3 fatty acid. So I'm not going to go through this table except to show you these are your common fatty acids. They have systematic names as well as common names. You can see here that you have oleic acid, which is a cis delta 9 octadecanoate, decinoate, excuse me. You have linoleic acid, which is an essential fatty acid. This is 9,12 octadecadienoic acid. And then you have alpha linolenic acid, which is an omega-3. All cis, again, double bonds, 9,12,15. See 15, 3 away from the methyl terminus because the 18 carbon fatty acid. Octadecatrienoate. Those are some of the structures. Okay, give you an idea of common name for fatty acids, how many double bonds they have, systematic names, and then there's the formula just in longhand. So, pathway for the fatty acid to become desaturated is first elongation. So you have an acyl-CoA reacts with acetyl-CoA to elongate it. This enzyme is called beta-ketothiolase. You've now extended this fatty acid 2 carbons more. See? Then you have a dehydrogenase using NADH. Then you make this intermediate. You go through the enyl-CoA dehydratase where you remove water. You generate double bond when you remove water here. Say there's the OH group, one of those hydrogens, prohydrogen. Then ultimately you get the reductase, and you make the next elongation product. So C16, C18 fatty acids in the diet are elongated first, right? Now this is outside the fatty acid synthase complex. These are elongase reactions that can occur in organelles like ER mitochondria. Okay. Then you start pumping in double bonds. So the stereoCoA desaturase, remember it's way down here, and that's the making the oleic acid, so the middle of the molecule. But you also have a so-called delta 6, delta 5, and delta 4. Again, these are moving closer and closer to the carboxy terminus, which is a sterified to CoA, or is an oxygen ester to a phospholipid during desaturation. Okay, so thioester or oxygen ester. That's the ones that can supposedly occur in humans. You're not able to put double bonds towards the methyl terminus after that middle of the molecule, the 9 position, for example, because we do not have the delta 12 or the delta 15 desaturases. We don't express them in our cells. That's why linoleic and alpha linolenic acid are essential in our diet. We get those from microbes or plants, right? Or from animals that have eaten a correct amount of those microbes or plants. It's just showing you the same pathway in the diet. Then you take in alpha linolenic acid. There's your delta 6 desaturase. It gets elongated here to 20 carbon fatty acid. And ultimately, the next desaturase, the delta 5, will give you the eicosapentanoate. Eicosapentanoate then gets elongated, the 22 carbon fatty acid. There used to be believed that there was a delta 4. There actually isn't. This actually gets elongated once more, and then it's basically a beta oxidation after you pump in the last double bond using another delta 5. And you don't need to worry about that right now. Looking at linoleic acid shown here, you get, again, the same desaturase. You get an 18 carbon fatty acid with 3 double bonds, but notice it's 6,9,12. That maintains the omega-6 configuration, a geometric configuration, right, as well as the positional configuration, because it's a cis double bonds. Like this 20:3 intermediate, that's an 8-11-14, and then again, delta 5 desaturase makes arachidonic acid. 20:4, double bonds with the 5, 8, 11, and 14 positions, so it's 6 carbons away from the methyl terminus, so it makes 6 fatty acid. Here are the two major ones then, icosapentaenoate and arachidonate, and also we make a lot of DHA in the central nervous system, but the delta 4 desaturase is necessary because you elongate, you go through limit beta oxidation after you've added the double bond, and you end up with a double bond in the right position, so you get 22 carbon fatty acids with 6 double bonds. And those are at 4, 7, 10, 13, 16, 19. And that's DHA, docosahexaenoic acid. DHA and EPA are what are known as marine lipids or oil or, what are they called? Marine oil lipids. And that's what you buy at the grocery store or at the Walgreens. And this gives you an enrichment of omega-3 fatty acids. But you can synthesize them de novo yourself from microsomal enzyme shown here, as long as you have the sufficient amount of enzyme expression, the cofactors necessary, such as nucleotides, NAD, NADH, FAD, FADH2, things like this, but also absorption is necessary, right? And the correct diet. Just showing you exactly the same pathway again, just maybe it's a bit easier to see. These are the desaturase enzymes. There's the delta 6. Here's this chain elongation I told you about. So you've got, for example, icosapentaenoic acid gets elongated to this docosapentaenoic acid, gets elongated again to this 24 carbon fatty acid, all maintaining omega-3. Then a delta 6 comes in, pumps in that last double bond, then you remove 2 carbons and you're left with 22:6. So not a delta 5, as I mentioned, that was a mistake. It's a delta 6 there at the end. Same thing can happen in the omega-6 series too, by the way, but primarily that doesn't happen. Most of what you make in the human is this arachidonate shown here. All right. Now, there's this sterile response element binding protein that normally exists on the endoplasmic reticulum, and insulin or TNF alpha, which is a pro-inflammatory cytokine, can induce a proteolytic degradation so that this SREBP1C is released from the ER, moves through the Golgi apparatus, and actually makes it through the nuclear membrane and into the chromatin, where it will bind to a sterile response element. So this has to do with a depletion of cholesterol in the endoplasmic reticulum and the Golgi, and the nuclear envelope hardly has any, that will allow for the vacant opening of the protein to be attacked by the proteases. So the proteases are activated by things like insulin, necrosis factor alpha, but also a depletion of sterol in those membranes will result in the same thing. In fact, they're complementary and co-migrating in terms of expression patterns of the SREBP in this final form into the nucleus. And then what happens when you get this transcription factor, that's what this was, at the ER, then the Golgi, then into the nucleus, into the chromatin. And it binds to this is basically a cis element in the DNA, promoter region if you like. You'll increase the amount of acetyl-CoA-carboxylase, rate-limiting step for de novo fatty acid synthesis. You'll increase the amount of fatty acid synthase, right? That's the polyprotein itself, which will make palmitoyl-CoA, and then free palmitate after the thioesterase reaction, all part of the FS complex. And also it'll increase the amount of glycerol-3-phosphate acyltransferase, so you make glycerolipids. The sphingolipid metabolism is also enhanced this way. So again, this is showing you a little bit about what's going on in the muscle versus the liver. Insulin binds to the insulin receptor. That then activates the insulin response substrate, or the IRS, 1,2 it's called. That will activate phosphodiesterol 3 kinase, which will then activate the protein kinase B, and that will enhance the amount, you see here, this will enhance the amount of GLUT4, and GLUT4 gets into the membrane and the muscle tissue and allow for glucose uptake. Now, if there's a lot of free fatty acid around because of the obesogenic state, that will induce protein kinase C theta, which will inhibit insulin signaling and therefore inhibit the movement of the GLUT4 from endosomal compartments to the plasma membrane. Free fatty acids also directly inhibit GLUT4 transport while they're whatever's localized to the plasma membrane in muscle. This will increase the diabetic state because you have more circulating glucose, right? Because of the higher level of free fatty acid, because of the lipogenic state. Remember adipose level lipase reactions. In the liver, you also get some insulin activity working through the IRS, and it turns on again phosphatidylinositol trisphosphate kinase, which will turn on PKB. Now, what that does in the liver, though, rather than moving a GLUT transporter, because the liver doesn't need it, is it decreases gluconeogenesis and it increases glycolysis, right? Because you're getting in all this glucose, presumably. But when you have high levels of free fatty acid, like in the obesogenic state, what happens is that you fire up PKC theta, same enzyme, the protein kinase, theta isoform, that will again block the insulin response substrate 1,2, so you don't get this pathway, so this gets corrupted. But rather than that, you also get a corruption of glucose uptake even in the liver, because GLUT2 is just a sodium transporter, right? So sodium transporter, so you actually get hyperglucosemia because of this increase in free fatty acid, which blocks the normal insulin signaling, either in the muscle where it's more predominant, and also in adipose, not shown here, but also in the liver. But in the liver, the insulin's not involved in glucose uptake, but it is involved in gluconeogenesis and glycolysis, and here you get this dysregulation. So what happens here is you get an amplification of gluconeogenesis because this is stopped here. So rather than this arrow going down, it goes up. Right? So you get more sugar synthesis, more gluconeogenesis, enhancing the diabetic state. But you can see that the reason that I always say that diabetes is dyslipidemia, it's the fatty acid that's causing the problem, not the glucose. Fatty acids cause. Now, the glucose can cause problem in circulation. It can cause glycosylation from hemoglobin, for example, decrease the amount of oxyhemoglobin, all that. However, metabolically, it's the fatty acid dyslipidemia, which is inducing the diabetic state, in particular the type 2 diabetes associated with obesity. Okay, that's what I'm showing you here. Now we're at the point where I could say dyslipidemia, high circulating fatty acid, is common in obesity. Fatty acyl-CoA metabolism blocks glycolysis in the liver by increasing ATP production. Okay, and that's through the NAD and FAD reduction, of course. So you get a massive amount of beta oxidation, so you get a massive amount of NADH and FADH2, which will run through the electron transport chain, and then the proton-electron motive force will generate more ATP, right? So high NADH also blocks pyruvate dehydrogenase. Remember, that's allowing for glucose-derived carbon to enter into the mitochondria via the pyruvate dehydrogenase to make acetyl-CoA. But it will also block the glycolytic enzyme glyceraldehyde dehydrogenase, glyceraldehyde 3-phosphate dehydrogenase, and that further will inhibit hepatic glycolysis, which we talked about in lecture a while back when I was talking about glycolytic regulation. It was around Christmas time I gave you that video lecture, I think. So low glucose utilization increases serum glucose, as I showed you in that last cartoon diagram. That increases pancreatic insulin secretion, finally leads to insulin insufficiency because of excessive induction of insulin secretion. You start to get to degeneration of the beta cells of the pancreas, right? That's where the insulin is secreted from. And so you get insulin desensitization, right? Or insulin resistance, it's called. And you get, before that, you get hyperinsulinemia. Then you get hypoinsulinemia because the total amount of insulin secreted, even in type 2 diabetic, later stage, highly obese, older population, is actually an insulin insufficiency overall. At least that's the phenotype that's generated. Even with high circulating insulin is still possible, even in later stages. So again, this is giving you a really nasty formula, right? You're overweight, you become insulin resistant, right? Because of that fatty acid effect. Free fatty acids turning on TNF-alpha. I just showed you what that does in the muscle and the liver. You get metabolic syndrome because of that, the hypertension. That's why we went through the kidney pathology. Metabolic syndrome is going to lead then to type 2 diabetes, high level circulating glucose, hypertension, obesogenic environment, high levels of circulating glucose, lack of glucose uptake because of insulin resistance. Same time, glucose-free fatty acid and insulin are going to cause the pancreas to shut down. It can no longer compensate by getting hyperinsulinemia. Finally, you're going to shut down because of basically beta cell degradation, destruction, because of the high level of signaling from free fatty acid and glucose, causing glycosylation of proteins that normally act as transcription factors to regulate insulin synthesis, secretion, and of course conversion, the convertase reactions. Free fatty acid are all causing that problem. So you get initially in metabolic syndrome, you get elevated insulin, hyperinsulinemia, you get blood sugar levels are still normal in metabolic syndrome. That's why it's not diabetes, since you restrict them. But you do have high levels of circulating triacylglycerol, not triglycerides. I'm sorry, this came from a textbook. There are not 3 glycerol compounds. It's 3 acyl groups. Triacylglycerol gets elevated. High-density lipoprotein levels are decreased because of the lack of transfer of lipids from the IDL, LDL, VLDL fraction to the HDL, coming back in circulation and going to the liver. So that gives you high blood pressure. You get high blood pressure, again, because of chronic damage to the glomerula of the kidney. That will then induce hypertension because of its effect on salt absorption and salt uptake. I didn't show you that, but I did in previous lectures, I know. Ultimately, when you get type 2 diabetes, you get insulin may be higher, but it's not functioning any longer. Blood sugar levels are really high, so that's what's called diabetes. You see, it's not really not because of the sugar. That's a result, not an induction, right? Even counting this in, and the glycosylation reactions, even counting against the free fatty acids, causing the problem. Because there's an excessive amount, it doesn't go down because of the slow level of circulation, because it can bind to serum albumin and of course the lipoprotein fraction, even via TAG, right? And the lipase. So you get high levels of sugar. That's the diabetic, as I said. High levels of triacylglycerol again. You still have low high-density lipoprotein, and you get high blood pressure also with type 2 diabetes. And you get all this inflammation, and this inflammation results in all those other diseases like NASH, we talked about, NAFLD, hepatocellular carcinoma, and then various other kinds of neurodegenerative steps, even the central nervous system. All right. So here's a paper published in 2013 in a journal called Intech Open, which I published in. Not a bad journal. It's free and open. And in fact, you can read this paper without going through a paywall. Here's the aging component, okay? And that's what I'm going to leave you with today, because it's going to be a 2-part video lecture. Come right back to tomorrow with part 2. So the aging process is going to cause what's known as a neurodegenerative complex in this paper. You're going to get proteinopathies because you're going to get protein aggregation. Some of it has to do with the transglutaminase induction. You also get loss of neurons because basically of necrosis, right? Neurodegeneration occurs. This is all associated with aging. Obesity enhances the activity of the aging chronological disruption of gene expression and the pathophysiology and the morbidity associated with the increase in reactive oxygen, reactive nitrogen, etc. Closing of vascular beds, causing hypoxia and the wrong kind of organ, leading to neurodegeneration, for example, or sometimes a neuroblastoma or a glioblastoma, then resulting in brain cancer. That's another thing that increases. That's a pathology increases with aging, of course. But pro-inflammatory response because of aging, oxidative stress because of all the reactive oxygen, because of the lack of complete reduction of molecular oxygen during electron transport chain activity. You start losing growth factor expression because of the inappropriate splicing mechanisms, gene expression because of the dysfunctional, the aggregated dysfunctional chromatin remodeling complex because of what I just described to you a few moments ago, but also, so you remember the acetylation and the methylation and the propionylation and the ubiquitinylation of chromatin and of histones, of lysine residues on histones because of the free fatty acids, because the acetoacetyl-CoA increases, right, because of all the acetate. So you get a full-blown alteration of transcriptome, proteome, lipidome, right? That's what we're saying here. And that will then corrupt growth factors and hormone regulation. So you get an age-related neurodegenerative complex, and then in the central nervous system, then the presentation of that in the elderly, which is no surprise because of the aging and because of obesity, if it's chronic throughout life, mood changes, memory losses, and movement dysfunction. And that all had to do, remember, with the central nervous system. That's why I went through all the clock genes, and I discussed that going, again, way back from Halloween through Thanksgiving and Christmas and into the new year. So hopefully that will give you some idea where we are in this long arc of dysfunctionality that's linked to obesity and how that relates to pro-inflammatory responses, which are already increasing because of the aging phenomena, ultimately leading to all the morbid diseases that occur in the aging or in the premature aging because of this obesity, because of chronic inflammatory pathways, including secretion of pro-inflammatory cytokines, chemokines, etc., leading to then higher levels of morbidity at younger ages, leading then to a whole constellation of very dangerous florid diseases, which can lead to mortality. Okay. So I'm going to leave you with that, and I'm going to get back to part 2, which is going to be interesting because I'm going to go into in depth then a couple of brand new papers, and we're going to talk about another aspect of this. I'm not going to tell you what it is because it's just going to confuse what I've already talked about today. Not confuse, but it'll add on too much. But ultimately, I'm going to leave you with this video lecture. I'll come right back with part 2, probably tomorrow, which would be the 10th of February, 2021. So this is Dr. Dan Guerra from Authentic Biochemistry Studios giving you this video lecture saying bye for now.